Hello everybody, and thank you for choosing to watch another video on qualitative and quantitative analysis of pharmaceutical excipient. Today I'm going to talk about lecithin, and I'm calling it part 2. In part 1 I did talk about lecithin and did the quantitation and the study of this excipient using GC, and now in part 2 I'm going to show you data on HPLC with ELSD detector. Very briefly, what is lecithin? Lecithin is a mixture of different organic molecules. It is a cocktail of phospholipids, glycolipids, triglyceride, and other stuff. And in this slide, we can see the composition of each groups in a liquid lecithin. Now, lecithin comes in all different forms, and it is a natural product, and it can be from plant-based or animal-based. There is a very nice publication which is in Cosmetic, a journal of Cosmetic. It reviews different lecithins from different sources. It shows the composition of the PC, which is phosphatidylcholine, or PE, phosphatidylethanolamine, and other phospholipids. The lecithin, it is not only differ in its source, and also they are different when they are processed. So we have a crude lecithin, or call it liquid lecithin, and that can be purified and turned into, let's say, de-oiled lecithin. And the process of de-oiling is that they will remove everything that is soluble in acetone, and the end product is something, a type of lecithin which is solid and is enriched by phospholipids. And also it can be further purified or processed and turned into something with high phosphatidylcholine content. So we have lecithin that are different by their source, or animal-based or different plants, and they are also different by their process. About the benefit of lecithin for skin, there's a lot of information out there, and they make a lot of claims, or they give information about their benefits. They talk about it is keeping skin hydrated or moisturized, protective barrier, is a good emulsifier, and all this stuff. I don't want to debate them, but I can tell you my opinion. The fact is, lecithin doesn't like water, and so if you apply lecithin, which is very gooey stuff, on the skin, it's not going to provide any moisture to your skin. It will probably prevent moisture from losing from the skin, but it's not going to hydrate it. It is an emulsifier that is mainly used in water oil formulation, or for formulation that has a lot of oil. It is soluble in oil, but it doesn't like water. It is natural excipient, so that's a good thing about it. A lot of emulsifying agents are synthetic, and maybe they are not compatible with our skin. But this stuff is actually produced by our body. And also, I want to say that this excipient is very hard to work with. It is not very soluble, definitely not soluble in water. It is poorly soluble in fat phase, and it is a different excipient to work with. These are my opinion about this excipient, and everybody knows that this is not very popular emulsifying agent, and is not used in a pharmaceutical product a lot. So for the qualitative and quantitative analysis, I'm going to talk about normal phase chromatography with HPLC with ELSD detector. And as I said before, in part 1 I talk about GC with FID. I very much like to start with the method in USP NF. This method is in recent publication of NF, and it was absent in the previous one. I don't know at what point they started offering this method, but it is a very nice method with ELSD detector. and it is for raw material testing. I have to admit that I've never used this method. The main reason is that it requires to have the column, which is I'm trying to look, is L20 column. L20 column is a dial, is a special type column that it is silicon, and they put probably propylene glycol, something similar to propylene glycol, on the stationary phase. I didn't have this column, and it was kind of looked pricey to me, so I didn't go to purchase one. Maybe in future I will, when I have this type of column and I will generate some chromatography, I will talk about it. But so this is one method that probably would be a starting point for anyone who has L20. It is for raw material testing, and probably you need to see if it's applicable for your application. There is another method by Agilent. This one is normal phase, and it uses silica column. Mobile phase is IPA, hexane, water with ammonium hydroxide, and they are using the Agilent ELSD detector. I have tried this method with mixed result. I did not get a proper specificity with this method, and I had my excipients in the formulation correlating with the PCs, or peak of interest. So most of the time I had did not have a desirable chromatography that I was looking for. But this is one method that people want to try. The other method is published by Thermo Fisher. Again, this is a normal phase, and as I recall, they have two conditions, one for analytical column, one of them is for prep column. Mobile phase is a combination of 2-propanol, isooctane, and the mobile phase A is diethylamine formate buffer at pH 3. Once again, I had mixed result with this chromatography. My condition, because I'm not using exactly the Thermo Fisher conditions, my detector generated a lot of noise, and I could hardly see anything. So I did not get desirable chromatography with this method. Maybe if you have the Thermo Fisher equipment with the columns, and if you exactly follow their method, you will get the chromatography that they are showing on this picture. The next one is the publication in Dairy and Veterinary Science journal. It is the determination of lecithin in milk, and I have tried this method, and I got the very good and satisfactory chromatography. Here in this publication, they show a chromatography of lecithin in the raw cow milk, and I was able to generate similar chromatography, but not exactly using their condition. Now in this slide we see the chromatography that actually I generated using different column. It is a normal phase column, it is a silica column, but my equipment is not exactly as what they describe, and the mobile phase gradient program is not exactly the same. But I managed to generate very nice chromatography. We see phospholipids in 20 minutes range, all 4 of them. You get a PE, PS, PI, and a PC phospholipids. Also have this minor peaks, which I believe, but I'm not so sure, that they are assigned to glycolipids, and also the triglyceride eluting at 3 minutes. This is a chromatography of lecithin. Is a liquid lecithin. I don't want to call it raw lecithin, but it is a liquid lecithin, that you have all these components in a lecithin. The next chromatography is the chromatogram of deoid lecithin. In the case here, we see that the triglyceride peak is absent, and the amount of the phospholipids are significantly higher than any other components. So I'm very satisfied with these ELSD HPLC conditions, and I will thank this journal that gave me a starting point to develop this method. Before I leave, I would like to show this publication that's actually for chromatography of glycolipids, not lecithin, and this chromatography actually is very useful for assigning your peaks. Here are some of the references, the USPNF for sure, and other publications, but Thermo Science, Agile, and Ferrero Group. Thank you very much. I appreciate if you can write a comment. If you had experience working with lecithin, I really like to hear your experience. If you like this video, please push the like button, and of course I appreciate if you subscribe. Thank you very much and have a good time.